Okay, in this video tutorial we're going to talk about DNA replication. And this has always confused me a little bit. I don't know why in the Cambridge curriculum DNA replication actually falls under genetic control. I would have thought it would make a lot more sense to put it under the nuclear division. Especially since one key thing that I need to say now is students often get confused between transcription translation, so that's gene expression, and DNA replication, whereas they're fairly independent events. So to be honest, most cells actually sort of greatly reduce their gene expression, stop expressing most genes during replication because it's such an intensive process. So I often make the analogy to my students that DNA gene expression is really your day-to-day work, the cell doing its day-to-day job, its business, and DNA replication is like giving birth. And I say to especially to my female students, when you're giving birth, it's unlikely you'll be working at the same time. So DNA replication is really replication simply means to copy. So it's all about how does a cell copy its DNA. And let's talk, before we get into that, let's just talk quickly about why does a cell need to copy its DNA. Well, DNA replication is an essential step that kind of precedes cellular division. So in order for a cell to be able to divide, it must first copy its DNA. And so for growth and repair, we need to make copies, we need cellular division, which relies, as I said, on DNA replication. So DNA replication is often said to be semi-conservative. So semi-conservative, if you think about that, really kind of basically translates to semi being half, like a semicircle. So semi-half, conserve, to conserve is to save. So if you conserve water, you're saving water. So it means half saved or half kept. And what we're essentially saying here is that we start off with a DNA strand. So we'll have my DNA here. And essentially what happens is that the strand is split into two, and then each half, so these strands are split into two, and then each of those strands is used as a template on which a new strand can be synthesized. And this was, even before it was before we had evidence, this was the most likely candidate. It was the suspected mechanism of DNA replication because of the base pairing. The double-stranded nature of DNA was already a clue that this might be the mechanism. So scientists kind of assumed before we had evidence that the DNA would be split into two strands, and each strand would be used as a template, and that simply by matching, by base pairing the correct nucleotides onto the original template strand, simply by base pairing the correct nucleotides on the template strand, you could make the complementary strand. So that's essentially what happens. Nucleotides are paired up, matched. The bases are matched to ensure that the strand, the opposite strand, is complementary. Sorry, a bit of background noise there, I know. Anyway, so you can see here the original parent DNA. So here we've got a blue strand of the parent DNA, and a red strand is also parent DNA here. And then it's split into two, and a new strand is synthesized that's made from nucleotides that are simply matched up to the bases or the nucleotides on the original DNA. So this is said to be semi-conservative, because if you look at the new strands of DNA, so eventually this will completely unwind and we'll have two complete new copies. The new DNA is actually made up of half old DNA, so one strand the original DNA and one strand new DNA. So we say the semi-conservative process of DNA replication is when DNA is split into two and each half is used as a template to make its complementary strand, the other half. The complementary strands are made using free or spare nucleotides that are floating around the nucleus. And again, a lot of students ask, well, where do these nucleotides come from? So again, they can either be synthesized, or I assume that they may also come from our diet. We eat things with DNA in them all the time. So it's very important, essentially, that a cell's machinery matches the bases correctly as it's making the new strands, so that the new copies will be identical. And if they're not, that's when we say a mutation has occurred, and that can have serious implications on the function. So as we looked at in the last videos, if we change the sequence in the DNA, that could change the sequence in the mRNA, which would change this could change the sequence of amino acids, which could change the protein's structure and function, and that might affect one of our characteristics. So let's get into it in a little bit more detail. So I'm going to go through some steps here, and I'll talk at the end. I'll do a quick little summary. Now you probably don't need to know it in quite this much detail, but some students find it easier to have kind of the bigger picture. So the key thing here is it's unlikely that you'll be asked to kind of write an essay explaining the entire process. What's more likely is you may get questions in exams that ask you to kind of explain the function of for you to name and explain the function of some of the enzymes involved in this process, or they might name an enzyme and ask you to say, what role does it have. So there's a few key terms here that I've often coloured in at the start of each of these points. Sorry. So essentially, the first step is that the strands must be separated, and this is done by an enzyme called helicase, or the Americans will say helicase, and this essentially breaks the hydrogen bonds that holds the two strands together. So the splitting usually starts in a place that's rich in AT bonds, because between an adenine and thymine, between an A and a T, there are only two hydrogen bonds. So helicase basically splits the two, and this is called a replication fork. So you can see not a fork like one that you eat with, but we often talk about a fork in a river. So like here's the handle, and here it kind of splits. Well, I guess I'm referring to the fork you eat with, but like when a river forks or splits, we say that's a fork in the river or a fork in the road. And we would say that this replication fork is moving in this direction. So the helicase is splitting the bonds, so it's moving here, making the arms kind of larger as it splits the strands. So we'd say the replication fork is moving in, to the left in this case. So let me get rid of my arrow. Okay, so the next step is that we need to make a little starting flag. Is the best way I can describe this. So RNA polymerase, and if you remember, RNA polymerase is the RNA piecing together enzyme. It's also sometimes referred to as RNA primase. So this will make a short complementary sequence, just a very short one, that's complementary to the start of the DNA here. And so it basically it provides a starting point for DNA replication. So on the leading strand, so we call one side of the DNA the leading strand and the other side the lagging strand, and we'll look at why they have those names in a second. So just bear with me for now. The top strand here we're going to call the leading strand, and that's the one where DNA polymerase can now attach onto that primer and make a copy of the DNA, in a 3 to 5 prime direction. So I should be clear about that. It's reading the DNA, so it's reading the strand in a 3 to 5 prime direction. So the strand it's actually making will be opposite to that. So it'll be adding nucleotides onto the 3 prime end. It'll be running 5 to 3 prime. So DNA polymerase, and that's worth noting, DNA polymerase can only read DNA in a 3 to 5 prime direction. Okay, which means it makes or synthesizes a complementary strand that's 5 to 3 prime. So DNA polymerase can only read in a 3 to 5 prime direction. So that's not an issue on this strand, where it can attach onto the primer and it can move from this 3 prime end, read along here, moving steadily forward towards the 5 prime end. And as it moves in that direction, the reason that this is not an issue is as the DNA polymerase moves in this direction, helicase is steadily opening up these or breaking these hydrogen bonds so that it can continue just to zip along the strand And so if you look down in the next diagram, for instance, helicases opened up the two strands a little bit further, and essentially now the polymerase can simply continue along that strand in one kind of continuous motion. So on the lagging strand, things are a little bit more complicated. On the lagging strand here, DNA polymerase must start at the 3' end. So the 3' end, or at least the end that's most 3', closest to the 3' end, that's exposed would be over, sort of over here. So an RNA primer is added here, and then DNA polymerase starts to copy that. So I'll draw kind of a quick series of just small diagrams. So I guess what I'm trying to show here is this. So helicase splits open the DNA, and on the leading strand it can just continue along. But on the lagging strand, what it has to do is it has to put a little primer here, and then DNA polymerase can jump on there. So this is the 3' end, and it can move towards the 5' end. So it can read the 3' end here on the original DNA strand towards the 5' end. It can zip along there and make a copy of this region. But while it's doing that, helicase is busy splitting open this DNA strand. So by the time it's finished, what we'll actually have will look more like this. Sorry, that's the wrong color. So while DNA polymerase is doing that section, helicase is busy unzipping the DNA. So what happens next is that essentially the primer was put maybe here, and DNA polymerase has done this section, but now suddenly this whole new section is exposed. So what happens is we need to put down another primer at the 3' end here, another little starting point. So it's an RNA primer, and then DNA polymerase can jump on and synthesize that DNA to fill in this gap. But while that's going on, of course, the helicase is busy splitting the hydrogen bonds that are in between the two strands here. And so while that's going on, by the time DNA polymerase has finished that, we will have opened up even more of the DNA. We will have now, we laid down that first primer, and we put, some DNA on the end of that, and then we laid down another primer, and we put DNA polymerase filled in the DNA there. So now when we're going to have to lay down another little RNA primer and fill in this gap here again. So the issue is essentially that the key thing here, and that's probably all you really need to remember, you probably don't need to be able to explain it in quite so much detail, is essentially that on one strand, the leading strand, DNA polymerase can synthesize the DNA continuously. Whereas on the other strand, because DNA polymerase, the DNA piecing together enzyme, can only read DNA from a 3' to a 5' direction, it has to copy the DNA or synthesize the complementary strand in short bursts as the DNA continues to be opened further. So here it'll do this bit, then it'll come back and by that point it'll have opened up further, it'll do this bit, and then it will have opened up even further, it'll come back and do this bit. So in that respect, I guess that's what this diagram is trying to show down here. So because DNA polymerase can only read DNA in the 3' to 5' direction, things are a bit more complicated. So as the original parent strands continue to unzip, new RNA primers must be added, giving DNA polymerase another starting point, and the DNA polymerase can be synthesized in short bursts rather than continuously. So this results in what we call Okazaki fragments. Okazaki fragments are essentially, we've got here on the lagging strand short sections of DNA and RNA. And Okazaki was basically a clever scientist who came along, and he heated DNA during the replication process. And what he found was that he always had, in replicating cells, that a portion, a portion of the DNA strands were long. So when you heat DNA, you can heat it sufficiently to break the hydrogen bonds, so to separate the strands. So we got some long strands, which are the original parental strands, the strands we started with. He got a medium-sized, piece of DNA, which was on the leading strand, and he got lots of short sections of DNA. And that was a clue that the fact that he got this one sort of reasonably long, wasn't quite as long as his other ones, and then lots of small sections, kind of was a clue that maybe it wasn't quite as simple as people thought, and that maybe replication was slightly different on the two strands. So we call this the lagging strand because it does lag behind a little bit. This one can carry on continuously, whereas here it'll be finishing off this bit, and this bit won't be copied until it comes back and fills that in. So it kind of lags behind every so often. It lags behind a little bit and then has to catch up. So step 6 here. So essentially the last step is another DNA polymerase, so another DNA piecing together enzyme, just a slightly different version of it. So there are actually a few different types of DNA polymerase, comes in and replaces the RNA primers, so that we've got nothing but DNA on the newly synthesized strand. So here I've just shown the lagging strand. So on the lagging strand, where we had those RNA primers, they're removed and replaced with DNA. So the very last step is basically this, is that there are still a few little nicks in the backbone. So sorry, if I go back, even though we've replaced the RNA primers with DNA, the backbone, the sugar phosphates in the backbone aren't connected. So we have a special enzyme called ligase, DNA ligase, which essentially induces a phosphodiester bond. Probably don't need to know that, but induces a bond to form in the backbone to connect together the backbone in that lagging strand, in the newly synthesized lagging strand. I've put a few details here about telomeres and proofreading, but you probably don't really need to know that, so I think I'm going to skip that at this stage. This is something I just briefly mentioned in class. You could pause the video, and if it's big enough, hopefully you can read that if you're really interested. So lastly, it's worth just mentioning here, this is that when we go into nuclear division, if you're looking at nuclear division later and mitosis, meiosis and things like that, DNA replication is essentially you have a single strand of DNA and it produces two strands. And these two strands in organisms that have chromosomes, so like humans and all animals and plants, this chromosome structure that you've often seen, that often has that sort of X-shaped, is essentially two DNA sausages. And these are, basically DNA in the form of chromosomes is generally only visible under the microscope in this chromosomal form when you've got these double copies. So shortly after DNA replication. So we don't ever really see the single sausage version, and that's because, truth be told, the single sausage version doesn't really sort of exist as such. It's at this stage, when we've only got one copy or one strand of the DNA, it's much more loosely packed. So it's more like, think like spaghetti in a bowl. It's not quite so compacted together. But during replication, when that strand is split into two and a complementary strand is synthesized, to help organize that, because we're talking about very long lengths of DNA, essentially we end up with a double copy of that piece of DNA, and those two copies are the two sausages, or chromatids are their proper name, that make up this X-shaped chromosome. So the chromosomes you normally see in pictures are essentially a double DNA sausage. Each sausage is one copy, is the chromatid. So inside that we'll have one copy of the DNA, and in this other side we'll have an entire, we'll have made another copy. So you can think about it as kind of DNA starting off as a single sausage, even though you can't see them because they're not tightly packed into a chromosome. And that strand kind of splits and is copied to produce two identical copies of the DNA, which are held together as that double sausage, the two chromatids that make up this whole thing here is a chromosome. So just worth mentioning, the point in the middle where they're held together is called the centromere. So just a few terms there. So essentially there you go, that's DNA replication. Now I said at the end I'd just try and summarize that quickly for you. So I think that the really key things to remember, and especially if you're not going for kind of 100%, the really key ideas to remember here is DNA replication is semi-conservative. So that means the DNA is split and each strand is used as a template, and that means the resulting DNA is half old, half new DNA. DNA replication involves enzymes, and the key ones to probably remember are helicase, which splits the hydrogen bonds and separates the strands, and probably DNA polymerase. So DNA polymerase is the one that actually makes the new strand of DNA. That's the DNA piecing together enzyme. So DNA polymerase is what actually synthesizes the new strand of DNA. What else? Those are probably the two main key enzymes. Remember, Okazaki fragments might be worth remembering. That one is just the fact that on one of the strands, the direction, the fact that DNA polymerase can only read 3 to 5 prime, means that the DNA needs to be copied in short bursts. And that probably would see you through, to be honest. And just be aware that these chromosomes that you see, the double sausage chromosomes you see, consist of two copies, or chromatids, which each chromatid is simply a condensed, a coiled up version of a copy of the DNA. So I think that's basically it. In the next video we'll look at some evidence for the semi-conservative model of DNA replication, and then we'll move on to nuclear division. We'll get into the different types of cell division that all start with DNA replication. Cool, so I hope you found that helpful.